Deep inside the basement of almost every major hospital in the world, there are these thousands of tiny, dusty wax blocks.
Yeah, just endless rows of them.
Right. And they sit on these endless rows of shelves holding preserved biopsy tissues and tumors from past patients. And for decades, the deep genetic secrets trapped inside those blocks were considered completely unreadable.
Totally locked away.
Because the chemicals they used to preserve them essentially, superglued the molecular structure shut. But today, we are going to talk about how that vault has finally been cracked open.
Which is incredibly timely, honestly. Because as you sit there right now, listening to this deep dive, there's a fascinating paradox playing out inside your own body.
Oh, this is the part that always gets me.
Yeah, so as we age, our cells are constantly dividing, right? And with every single division, they accumulate these tiny genetic mutations. They're essentially typos in your DNA.
Just microscopic errors copying over and over.
Exactly. Now, most of these typos do absolutely nothing. They're just harmless background noise. But a few of them, just a few, will quietly alter a cell's programming.
Setting off a chain reaction.
Right, a chain reaction that eventually sparks diseases like cancer or autoimmunity.
And the ultimate question in biology has always been, how do we know exactly what a specific mutation is making a specific cell do?
Right, connecting the typo to the actual behavior.
Exactly. So, welcome to this deep dive. Today, we are exploring this groundbreaking new technology called genotyping in Fixed Transcriptomes, or GIFT.
GIFT, yeah, it's a game changer.
It really is. Our mission today is to uncover how this method lets scientists look inside hundreds of thousands of individual cells, read their genetic typos, and simultaneously watch how those typos change the cell's behavior.
And incredibly, they can do all of this, even in those preserved decades-old medical samples we just talked about.
Which is wild, because for a long time, seeing both the cause and the effect at the exact same time was, nearly impossible.
Yeah, we could look at a mass of tissue and see that a mutation existed somewhere in there. But tracing that exact mutation to the specific abnormal behavior of a single individual cell, that level of resolution was just totally out of reach.
To give you a sense of why this is such a monumental leap, imagine trying to understand how a massive, bustling city works. Previously, scientists basically had two options.
Right, the addresses or the windows.
Yeah. They could read the addresses of the buildings, which is like sequencing the DNA to find where the mutations are. Or they could look through the windows to see what the people inside were doing.
Which is reading the RNA transcriptomes to see the cell's actual behavior.
But you could rarely do both simultaneously.
Almost never.
You couldn't say, the person at this specific address is doing this specific activity. And even when you could match them up, it was usually only for a tiny fraction of the city, like just one neighborhood.
Right.
But GIFT is basically this high-definition satellite system that maps the entire city at once. It tells you who lives at what address and what they are doing at the exact same time.
And if we look at the older technologies that tried to do this, methods like RT-based genotyping of transcriptomes or GAT, they were just severely limited by their underlying mechanics.
Because they relied on reverse transcription, right?
Exactly. Think of reverse transcription like trying to read a tape measure by starting at the very end and spooling it backward.
Okay, spooling backward.
Yeah. But if the RNA strand is broken or degraded, the tape measure just snaps. You can't read any further.
Oh, wow. So you just lose all the data past the break.
Exactly. Because of that physical limitation, those old methods could really only look at maybe 1 to 3 mutational spots per cell, and mostly just mutations that happen to sit right near the tail end of the RNA.
So if your mutation was buried deep in the middle of the transcript, you were completely out of luck.
Completely out of luck. And you also needed fresh, perfectly intact living cells for it to even work in the first place.
Which you definitely don't have with decades-old wax blocks.
Right. But GIFT completely bypasses those limitations.
So, okay, how do we build this new satellite? Before we start mapping the whole city, we need to understand the new camera they are using.
A molecular magic trick.
Exactly. Let's unpack that. How does GIFT read a broken transcript without needing that reversed transcription tape measure?
Well, it really comes down to this highly ingenious use of single-stranded DNA probes.
Okay.
Instead of trying to read the whole RNA strand from end to end, GIFT uses 2 rationally designed probes that act like highly specific magnetic strips.
Magnetic strips, got it.
Yeah, and they bind directly to the RNA transcript right where the suspected mutation lives. But the clever part is that they leave a tiny, deliberate gap. just a few nucleosides wide directly over the exact spot where the genetic typo is located.
Oh, I see. It's like placing two bookends on either side of a suspected typo in a book and leaving a blank space right over the typo itself.
That's a great way to picture it.
But with one crucial difference, right? These bookends are magnetic, designed to snap onto only one specific chapter of the genome.
Exactly. And once those magnetic bookends are locked in place, the system has to fill that blank space to actually read the typo. So it's called the gap fill reaction.
The gap fill reaction, okay.
But to make this work, the research team had to hunt for an enzyme that could perfectly fill that gap without, messing up the bookends.
Which sounds tricky.
Very tricky. They surveyed this massive landscape of polymerases and eventually found their ideal candidate in an enzyme called sulfolobus DNA polymerase V.
Whoa, hold on. Sulfolobus DNA polymerase V. That is quite the mouthful.
It is a bit of a tongue twister.
Why that specific one? Like, what makes an enzyme perfect for this gap fill reaction?
So it's actually because of what it lacks. It is missing two very specific biochemical traits, strand displacement and exonuclease activity.
Oh, okay, you lost me. Exonuclease activity. Why is it a good thing that our enzyme is missing pieces?
Well, let's go back to your bookend analogy. If the enzyme had strand displacement activity, it would act like a bulldozer.
A bulldozer.
Yeah, as it tried to fill the blank space, it wouldn't know when to stop.
Yeah.
You would just keep driving forward and push your second bookend right off the RNA strand.
Oh, man, so it ruins the whole setup.
Exactly. And on the other hand, if it had exonuclease activity, it would act like a molecular shredder. It would literally chew up the bookends from the edges before it even filled the gap.
Wow. Okay, so we need an enzyme that isn't a bulldozer and isn't a shredder.
Which is exactly what sulfolobus pol 4 is. Because it lacks both of those traits, it acts like a precision paving machine.
A precision paving machine. I love it.
Right. It patches the pothole between the probes, reads the typo, and then simply stops. It leaves the bookends completely intact.
So that makes total sense. It's precision engineering at the molecular level. But reading through the study, it wasn't just the enzyme doing the heavy lifting, right? There was a chemical additive they threw into the mix to boost the signal, betaine.
Oh, yeah. Betaine plays a critical role here. You see, DNA and RNA have this tendency to fold up on themselves.
Like getting tangled.
Yeah, especially in regions that are heavy in guanine and cytosine, the G and C nucleotides. They form these sticky, tight, secondary structures that just block enzymes from getting in.
So they're physically in the way.
Right. So betaine essentially acts as a chemical untangler. It relaxes those sticky DNA knots, ensuring the enzyme has clear access to the gap.
That's fascinating.
And it's incredibly effective.
Yeah.
Adding betaine into the mix boosted their cellular yield by a massive 60%.
60%. Okay, so just to pause and make sure we are tracking here, and for you listening, we've got this beautifully optimized chemistry. We've got our magnetic bookends flanking the mutation, the perfect pothole patching enzyme to read the typo, and betaine acting as a detangler so the enzyme can actually do its job.
That is the core mechanism, yes.
But the true test of this technology isn't just getting it to work on pristine, fresh cells grown in a carefully controlled lab, right?
Not at all.
The real breakthrough is applying this camera to those dusty wax blocks we talked about at the very beginning, unlocking the archives.
And this is where we have to talk about formalin-fixed paraffin-embedded tissue, or FFPE.
Right. Because when someone has a biopsy or a tumor removed at a hospital, this is usually how it's stored. They preserve it in formalin and encase it in wax.
Which is great if a pathologist just needs to look at the physical shape of the tissue under a microscope.
It keeps it structurally sound.
Yeah. But chemically, formalin is an absolute nightmare for molecular biology. The fixative literally cross-links the RNA.
Cross-links it?
Yeah, it ties the molecules together into this rigid, degraded mess. Traditionally, this chemical superglue makes the RNA completely inaccessible to single-cell sequencing.
It's like trying to read a book where all the pages have been fused together.
Exactly.
So how did they get past that? I mean, superglue is tough.
The GIF team utilized this brutal but incredibly effective solution high-temperature decross-linking. They subjected the preserved nuclei to an 80 degrees Celsius treatment.
Wait, 80 degrees Celsius, that is basically boiling it.
It is extremely hot, yeah.
I would think that would just disintegrate the cells completely.
It sounds totally extreme, but it happens to be the exact temperature threshold needed to melt away those chemical crosslinks without snapping the molecular backbones of the RNA.
Oh, wow. So it melts the glue but keeps the pages intact.
Exactly. That single heating step restored transcript accessibility, increasing the RNA recovery tenfold.
Tenfold?
Yeah. Suddenly, Those tightly bound, decades-old transcripts were open for business, and the gift trobes could finally bind to them.
Okay, let me play devil's advocate for a second here.
Sure, go for it.
You've just essentially boiled decades-old degraded tissue. You send your probes in. How do you know the data you're getting back is actually real? Like when you amplify old genetic material, doesn't it create a ton of noise and false signals?
Oh, it creates a massive amount of noise.
Right.
Specifically, you run into this phenomenon called PCR template switching.
What's that?
Well, when you are chemically amplifying the genetic material so you can actually read it, the molecular barcodes that identify which cell a transcript came from can accidentally get swapped.
Oh, I see. Like luggage tags getting swapped at a chaotic airport, so the suitcase ends up assigned to the completely wrong passenger.
That's a perfect analogy. And in genetics, assigning a cancer mutation to the wrong cell destroys your entire analysis.
Because you think a healthy cell is cancerous or vice versa?
Exactly. So to solve this, the team didn't just rely on chemistry. They built a rigorous, 4-step probabilistic framework to filter out those swap tags.
Okay, wait. How does probability catch a swapped luggage tag?
Think of the algorithm like a highly suspicious bouncer checking IDs at a club.
Okay, a bouncer.
Right. The model looks at the frequency of the barcodes. If it sees a barcode that appears perfectly, say, 990 times on one cell's RNA, but suddenly pops up 10 times on a completely different cell's RNA.
It gets suspicious.
Very suspicious. It calculates the statistical odds. It recognizes that those 10 weird appearances are likely artifacts of the amplification process, the swap tags, and it literally throws them out.
Wow, so it just kicks them out of the club?
Yep. It calculates the exact probability that a gapfill sequence belongs to a true allele, weeding out false positives so effectively that they achieved over 99% accuracy.
99%. That's incredible. And they proved this on an actual human brain tumor, right? A glioblastoma sample that had been preserved in one of those FFPE wax blocks.
They did. By applying GIFT to that archival glioblastoma, they found specific mutations in the EGFR gene and the AXL gene.
Okay.
But because GIFT looks at the single-cell level, they could actually prove which of those mutations co-occurred in the exact same cells.
Rather than just guessing based on a blended average of the whole tumor.
Right. If you only have bulk sequencing, where you just grind up the tissue and read it all at once like a smoothie, there are mathematically 73 possible evolutionary trees for how that specific glioblastoma might have developed.
73? That's too many to act on.
Way too many. But GIFT filtered out the noise and reduced that down to just 6 possible trees. They essentially hit rewind on a static block of wax and rebuilt the tumor's evolutionary history.
Just taking a piece of medical history and watching it evolve in high definition, that is so cool.
It really is.
But, as amazing as it is to rebuild one tumor's history, solving a disease requires finding universal patterns across thousands of people. And to do that, you have to scale up.
Which is the next major phase of the study. The researchers scaled GIFT up to profile over 712,000 cells.
Wow.
From a massive cohort of 35 patients suffering from myeloproliferative neoplasms, or MPN.
MPN. Right. That's a type of blood cancer characterized by the overproduction of blood cells.
Exactly. And remember how we said the older tape measure methods maxed out at capturing maybe 1 to 3 mutational targets per cell?
Yeah, the spooling backward problem.
Right. Well, in this MPN cohort, GIFT captured up to 611 targets per cell.
Wait, 611 compared to 3?
Yeah, it's a completely different league. It found mutations no matter where they were hidden along the entire length of the transcript.
The leap in resolution is just staggering. Now, in MPN, the primary villain driving the disease is a specific mutation called JAK2V6710F, right?
Yes. The JK2 mutation is notorious in this space.
Yeah.
And because GIFT is so scalable, the researchers leveraged it to do something entirely unprecedented.
What's that?
They didn't just compare a group of mutated patients against a group of healthy patients. Because they were looking at hundreds of thousands of individual cells, they compared the mutated GK2 cells directly to the healthy wild-type cells floating right next to them in the exact same patient.
Oh, that is brilliant. You completely eliminate all the outside variables. It's the same patient, the same environment, the same immune system. The only difference is that tiny typo in the DNA.
Exactly.
So what did they find when they put them head-to-head like that?
They discovered something that fundamentally changes how we view genetic disease. We often think of mutations as an on-off switch.
Right. You either have the typo and the cell is cancerous, or you don't and the cell is healthy.
Right. But GIFT revealed an allelic dosage effect.
An allelic dosage effect, let's translate that for everyone, meaning the amount of the mutation physically matters.
Let's break it down. You inherit 2 copies of every gene, right?
Sure.
The researchers looked closely at monocytes, which are white blood cells involved in the immune response. They watched as the mutation status went from wild type, meaning both copies of the gene were completely clean,
Okay.
to heterozygous, meaning one copy had the type O2 homozygous, meaning both copies were corrupted.
And what happened?
As the dosage of the typos increased, the cell's inflammatory response, specifically its interferon gamma signaling, scaled up in a perfect corresponding gradient.
Oh, wow. Here's where it gets really interesting. It's exactly like a volume dial on a stereo.
A volume dial, yeah.
FT doesn't just tell us if the heavy metal music is playing in the cell, it tells us exactly what number the volume dial is set to. It proves that the physical amount of the mutated gene directly dictates the intensity of the disease state.
That's spot on.
Like one typo means the inflammation volume is at a 5. 2 typos mean the volume is cranked to 10.
And that constant blaring inflammatory noise changes the entire cellular environment, puts immense stress on the body.
Which brings us to the most dramatic part of the research, right? What happens when that blaring noise eventually forces a cell to evolve into something much deadlier?
Exactly. Because MPN isn't always static. It can transform over time into something much more aggressive, like acute myeloid leukemia, or AML.
And the researchers used GIFT to catch this transformation happening in real time in a specific patient.
They did. They basically used GIFT like a molecular time machine. They tracked a patient whose MPN was actively progressing into AML.
Okay.
And by performing single-cell lineage reconstruction, they separated the patient's cancer cells into distinct clonal tribes. Let's call them clone A, B, C, and D.
Tribes. I like that.
Yeah, and each of these tribes splintered off from the original cancer and acquired different secondary mutations.
So they're essentially tracing the family tree of the cancer as it branches out. How did these different tribes behave? Like, did they all do the same thing?
They behaved entirely differently, which is the critical insight here. You had the exact same starting cell, type of blood stem cell, but its marching orders changed completely depending on its mutational tribe.
Give me an example.
For instance, the clone that picked up a mutation called EZH2 was ramping up that interferon inflammatory response we just talked about. It became a chronic immune alarm.
Okay.
Meanwhile, a different clone harboring a ZRSR2 mutation was triggering an entirely different chemical fire alarm, the TNF-alpha signaling pathway.
Wow. So the exact same type of cell is launching completely different weapons based on which genetic typos it picked up along the way.
Exactly.
But reading the case study, there was one clone in particular, clone A, that held the real secret to the leukemia, right?
Yes. Inside clone A, the GIFT platform detected a tiny, incredibly rare mutation in a gene called NRS.
How rare are we talking?
So rare that standard bulk sequencing of the patient's blood completely missed it.
Completely missed it.
Yeah. To use a visual, it wasn't just finding a needle in a haystack. It was like finding a single misspelled word hidden inside a massive library of encyclopedias.
But why does that one misspelled word matter so much?
Because when the researchers looked at longitudinal samples from this patient over time, they saw that this exact NRAS clone, that microscopic hidden seed, exploded.
Oh, wow.
Yeah, it expanded rapidly and became the aggressive leukemic blast population that drove the acute leukemia.
So they literally caught the spark before it became the forest fire.
That's exactly what it is. GIFT essentially allowed them to map the subtle hidden branches of the tumor and predict what it would become tomorrow, rather than just reacting to the tumor they could easily see today.
That is just incredible.
We can now track cancer evolution at a resolution that was previously considered science fiction.
Okay, let's take a breath and do a quick recap of everything we've uncovered in this deep dive.
Good idea.
So first, GIFT is this incredible new technology that bypasses the limits of old reverse transcription. It uses magnetic bookends and a smart gap-filling enzyme to read both a cell's RNA activity and its exact DNA mutations simultaneously without shredding the transcripts.
Crucial point.
Second, it can literally bring archival FFPE tissue back to life. By boiling samples at 80 degrees to melt the chemical superglue and using a bouncer-like algorithm to throw out false signals, it makes decades-old tumor blocks readable again.
Unlocking the archives.
Exactly. Third, it scales massively. By looking at hundreds of thousands of cells in MPN blood cancer, it proved that mutation dosage acts like a volume dial, directly controlling how much inflammation a cell produces.
The allelic dosage effect.
Right. And finally, it tracks the hidden evolutionary branches of a tumor. It can find a single misspelled word like that NRAS mutation, predicting how a cancer will progress into leukemia before the clinical symptoms ever show up.
Which fundamentally changes the horizon of medicine.
How?
Because true personalized therapies cannot rely on guessing what a tumor is doing based on a blended average of cells. You need to know exactly which individual cells have which mutations and exactly how those specific mutations are changing the cell's behavior.
It's all about the details.
Yes. GIFT provides the ultimate high-definition map to guide hyper-targeted treatments in the future.
Which leaves us with one final, incredibly provocative question to think about.
Let's hear it.
If GIFT can map these subtle hidden mutations and show exactly how they change a cell's behavior in cancer, could we eventually use it on completely healthy tissue?
Oh, that's a wild thought.
Right. Could we trace the cellular origins of aging itself? For you listening, what hidden genetic typos are quietly accumulating in your cells right now as you listen to this? And what secret instructions are they giving your body?
Definitely something to think about.
Something to ponder until our next deep dive.